Okay, so we're going to get started. Alrighty. So thank you all for your kind attention, and I look forward to presenting for you. It's really an honor to be here today. The title of my talk is The Role Dietary Heavy Metals Play in the Epigenetic Inheritance of Autism and ADHD. So today's discussion will cover the findings reported in our most recent publication. In March of this year, my collaborators and I published a review article titled Higher Rates of Autism and Attention Deficit Hyperactivity Disorder in American Children. Are food quality issues impacting epigenetic inheritance? Let me first give you a little bit of background, because what I'm presenting to you today follows a sequence of research that has been going on for nearly 20 years. And like some of the other speakers, we've been working on this, and it just keeps getting better and better. So I worked at the Food and Drug Administration in the United States for 9 of my 16 years in the United States Public Health Service. While I was working there, I was investigating the mercury cycle as part of my job. It turns out there's often mercury residue in the manufactured food product known as high fructose corn syrup. In Europe, it might be known as isoglucose or glucose fructose. They have different names for it. I found the mercury in the high fructose corn syrup by accident in 2005. At the time, the USDA, which is the United States Department of Agriculture, had reported each American was eating on average 60 pounds of high fructose corn syrup per year. That's opposed to like 2 pounds of broccoli, right? So that's a lot of high fructose corn syrup. That seemed, I felt the potential mercury exposure were a danger to pregnant women, infants, and children. The FDA did not agree with me, so I retired early from my career on December 31st, 2007, to publish my findings of mercury in high fructose corn syrup. Since my retirement and the publication of those original findings, I've been busy working with my collaborators to follow the line of research I started at the FDA. So we're, long 15, 16 years later. Our line of research is now known as of dietary heavy metal co-exposures on gene behavior and child neurodevelopmental outcomes. So the problem of dietary heavy metal co-exposures has been finally identified by the United States Congress in 2 reports published in 2021. So the first report, published February 4th, was called Baby Foods Are Tainted with Dangerous Levels of Arsenic, Lead, Cadmium, and Mercury. And then the second report on September 29th was titled New Disclosures Show Dangerous Levels of Toxic Heavy Metals in Even More Baby Foods. So Congress got the data from cooperative food manufacturers. So you may now be wondering if the Congress has taken any action. Not yet. So they're asleep at the switch. Let's move on to the topic of nutritional epigenetics so we can better understand how heavy metal co-exposures, they're not just in baby food, they're in throughout the food supply, can impact child neurodevelopmental and health outcomes. Nutritional epigenetics, the phrase, was first coined in a review article about elements in food that could impact gene behavior. Hannah Landecker, the author of that article, suggested that these elements could be toxic and change the way genes behave. Nutritional epigenetics is now considered the field of study that examines the effects of diet or food on gene expression. Researchers in this field seek to explain how prenatal diet can alter how the switches work to influence gene behaviors. Switches turn genes on or off. So here's an example of a switch called a methyl group. Now, one way that genes turn on and off is via methylation switches, which depend on dietary patterns. What you eat determines how many methyl groups you're going to have. DNA methylation is an important regulator of gene expression. A methyl group, which is a molecule made up of 1 carbon and 3 hydrogen atoms, attaches to DNA and serves as a switch to turn a gene on or off from one generation to the next, for better or worse. Genes must turn on so the body can produce proteins important for survival. And what we eat, our diet determines if we have enough methyl groups in our cells to regulate our genes properly. This chart provides information on common methyl group donating nutrients, to include methionine, folic acid, vitamin B12, vitamin B6, and choline. Pregnant women are often encouraged to take a folic acid supplement to prevent birth defects. There are other dietary factors that can impact gene expression. In general terms, they are food insecurity, poor prenatal diet, and food quality issues. Food insecurity is the inability to access enough food to meet basic needs. It is associated with increased risk of birth defects associated with DNA methylation. Poor prenatal diet is the same thing as unhealthy diet and has been shown to affect DNA methylation patterns and contribute to the development of type 2 diabetes, attention deficit hyperactivity disorder, and early onset conduct disorders in children. Food quality issues vary from one geographic region to the next, depending on the country, food safety practices, and manufacturing and agricultural regulations regarding heavy metal and pesticide residues. Mercury in high fructose corn syrup is but one example of a food quality issue that can impact gene expression. Food colors are another food quality issue in the United States. Petroleum-based food colors, which are made from petroleum, which comes from the ground, have been around since before FDA was even formed as an agency, and they may contain mercury and other heavy metals. FDA certifies that these food colors by batch to ensure that the concentrations of inorganic mercury, lead, arsenic, and chromium do not exceed the allowable levels. So there's, up to 10 parts per million lead, 1 part per million mercury, and 3 parts per million arsenic allowed in specific food colors that are made from petroleum, which comes from the ground. So here we see some of the certified food colors and the allowable heavy metal residues, along with some of the ultra-processed food products where they may be found. In the U.S., these certified food colors are found throughout the food supply and may be sources of heavy metal exposure in the general population. In addition to food colors, some preservatives like sodium benzoate, citric acid, they have allowable heavy metal levels. If we are consuming ultra-processed foods, then we are likely getting numerous co-exposures to lead, arsenic, and inorganic mercury. I bought these here. This food supply is pretty clean. I had to search for these items. They weren't easy to find. Needless to say, in the United States, the food quality is not everywhere. Let's briefly review our first nutritional epigenetics model with a focus on mercury toxicity. What happens inside of mom when she eats the standard American diet, which consists primarily of highly processed foods, containing high fructose corn syrup and vegetable oils. In addition to being a potential source of inorganic mercury exposure, the consumption of high fructose corn syrup may lead to zinc losses. In this first nutritional epigenetics investigation, we focused our research on the metallothionein, or the MT gene, which is zinc dependent. When the MT gene is expressed, toxic metals like mercury can be eliminated from the body. If mom is zinc deficient, she will be less able to produce the metallothionein protein and unable to excrete heavy metals. Instead, she will be bioaccumulating mercury and lead and other heavy metals in their blood. She will pass these heavy metals on to her infant along with her DNA methylation patterns. To avoid zinc deficits, a woman must avoid consuming high fructose corn syrup, yellow number 6 and yellow number 5, especially during pregnancy. Children with autism and ADHD are zinc deficient, and because they do not have enough zinc to build the metallothionein metal carrier protein, they tend to bioaccumulate heavy metals in their blood. So the more of this and this that they're eating, then the more heavy metals they're accumulating. Symptoms of autism and ADHD are correlated directly with the head. We know this clinical trial data from numerous trials. The higher the heavy metal levels in blood, the more severe the symptoms. In 2012, we reviewed the literature again and revised our model in a new article published in the Clinical Epigenetics Journal. Few people understood the role of diet in gene modulation or the impact of heavy metal exposures in child neurodevelopment. To develop this model, we compared the food supply and autism prevalence in Italy to the food supply and autism prevalence in the United States. In our review, we found a difference in PON1 gene activity between the two populations, different diets. We also found autism prevalence in Italy was much lower. Here we have our revised nutritional epigenetics model with a focus on the PON1 gene. In this updated model, we see the PON1 gene is a critical gene in children with autism and ADHD. The PON1 gene is calcium dependent and can be suppressed by, guess what? Fructose, high fructose corn syrup. At the time of this review, high fructose corn syrup was not being sold in Italy due to trade restrictions. High fructose corn syrup had not even made it to the EU marketplace. It turns out consumption of fructose by humans can lead to calcium losses when magnesium intake is low. We know this because the United States Department of Agriculture scientists did that study. With losses in calcium, PON1 gene activity decreases, and lead levels may also increase because lead and calcium do a dance. With lower PON1 activity, organophosphate pesticide exposures become more toxic in children with autism and ADHD because their little bodies can't produce that carboxylase enzyme to sound these pesticides. So when organophosphate and heavy metal exposures are running through the body, the cells undergo oxidative stress and tissues die. In the case of autism and ADHD, brain cells are impacted by these exposures. So we know that organophosphate pesticide exposure is common because the U.S. Department of Agriculture tracks the levels of organophosphate pesticide residues in crops grown in the United States. How can consumption of high fructose corn syrup make organophosphate exposure more toxic? Well, we know consumption of high fructose corn syrup can lead to calcium loss in humans. We know PON1 activity is calcium dependent. So with less calcium, there's less PON1 activity. The activity of PON1 is impaired in children with autism and ADHD. This means they're more susceptible to the neurotoxic effects of organophosphate exposure, which incidentally Where do you think organophosphate pesticide residues are found more often than not? Wheat. So, high fructose corn syrup consumption is a factor in the development of autism and ADHD due to the associated calcium loss and impact on PON1 activity. So what else can modulate or suppress the PON1 gene to create more autism and ADHD? The PON1 gene can also be suppressed by environmental and prenatal exposures to mercury and lead, heavy metals. So here you're getting a double whammy. In the case of prenatal exposures to certain pharmaceutical drugs like statins, cigarette smoking, and alcohol, those also suppress PON1. With regard to age, in the case of age, babies have lower PON1 gene activity. These are sensitive populations, and so dietary heavy metal exposures are going to be much more toxic. The PON1 gene is part of the immune system with respect to the body's ability to detoxify pesticides. Boys and baby boys have lower levels of PON1 gene activity. Thus, autism and ADHD are more prevalent in males. Now that we understand some of the mechanism involved in autism and ADHD, let's look at the raw autism prevalence data for special education that we analyzed recently. So even though student enrollment has remained relatively stable over the last 15 years, autism prevalence in children ages 6 to 21 increased 242% in the United States between 2006 and 2021. Developmental delay increased 184%, and across all disability categories, demand for special education services increased by 10.4%. And this is all while student enrollment is flat. So that's scary. Let's look at autism prevalence today in Italy and compare it to the prevalence in the US and see what's happened between 2012 and 2021. Autism prevalence in the US increased 54.4% during this period, while in Italy, it increased 360%. What happened? Well, what happened was, in 2017, the EU sugar quota system ended, which meant there were no more restrictions on the importation of high fructose corn syrup to the European Union. Here we have a bar chart produced by Starch Europe, the trade organization for high fructose corn syrup, which is known as isoglucose in the EU. You can see the two short bars representing before the end of the quota and after the end of the quota. The tall bar is the total output of isoglucose in million tons expected by 2026. So what do you think will happen to autism prevalence in Italy as high fructose corn syrup continues? Consumption continues to increase. I'm just guessing. We don't know. But that's a huge percent increase for Italy. 360%. Wow. So this brings us to the topic of useful biomarkers in autism and ADHD. We do have them now. Clinical trial data all over the world show children with autism accumulate heavy metals in their red blood cells. The children exhibiting symptoms of autism most often have higher inorganic mercury and lead levels in their blood compared to their neurotypical peers. We know that dietary intake of foods containing heavy metals connect directly to the heavy metals in the child's blood. We also know for certain that children with ADHD accumulate lead in their red blood cells. So children that exhibit symptoms of ADHD will have higher blood lead levels compared to their neurotypical peers. We also know that the heavy metal levels in the blood correlate directly with the severity of the symptoms exhibited by children diagnosed with these disorders. All this has happened through the last 15 years, all this data through clinical trials. So we've covered most of what we wrote about in our newest review article, but let's talk about DNA methylation patterns that get passed on from one generation to the next. Prenatal inorganic mercury exposure may induce changes in the PON1 gene that persist into early childhood. Among male children, prenatal mercury exposures were associated with cord blood methylation at the PON1 gene locus and lower cognitive test scores in childhood. See how it's coming together? Because male children have lower PON1 gene activity, prenatal mercury exposure associated with cord blood methylation at the PON1 gene may explain why autism and ADHD impact boys more often than girls. Bosak et al. Found epigenome-wide associations of prenatal heavy metal measurements, lead, and manganese. Or is that, I don't know what that is, I can't remember. In blood of mother with cord blood DNA methylation and persistent DNA changes in mid-childhood. In a meta-analysis of epigenome-wide association studies involving cord blood, Newman et al. Found DNA methylation at birth strongly associated with development of ADHD. So prior to 2019, we didn't have a model for studying the adverse impacts of dietary co-exposures to low-level lead, cadmium, inorganic mercury, all that, and tricks and, lucky charms and all that stuff we got in the United States. All the current allowable limits of exposure to heavy metals in the international and U.S. food supply are based on the expected response to an extrapolated dose of exposure to a single element. Fortunately, studies are now being conducted to determine the synergistic and epigenetic effects of co-exposures. We can thank Dr. Zhao in China. He's ahead of the game here. So with the methylation changes leading to transgenerational epigenetic inheritance, we're still talking about co-exposures. We've got the consumption of the ultra-processed foods that provide all those nice heavy metals and then result in mineral imbalances, calcium loss, zinc loss, and then our genes don't work right. We can't excrete the heavy metals, and our PON1 is suppressed. And so then what we end up with is a buildup of heavy metals, and then we have oxidative stress, and this leads to changes in methylation patterns. And this then leads to transgenerational epigenetic inheritance, which then we have more and more with each generation. Autism and ADHD. So what can we do? Well, we can change the narrative. We thought the food supply was safe. We now know the food supply is not safe. We are uneducated as to the allowable contaminants in the food supply. We're uneducated as to what co-exposures to these contaminants will do to our bodies and our offspring. We are poisoning future generations by what we eat today. And until we get educated, we are literally unsafe at any meal. So I did write a book, Unsafe at Any Meal, and I used the content of that book to develop curriculum that I have tested in 3 clinical trials to help participants reduce their intake of ultra-processed foods and significantly increase their intake of whole organic food. So you can train people how to eat right. And we actually did measure biomarker data in one study, and we found that the subjects who participated in the intervention significantly reduced their ultra-processed food intake, they had lower inorganic blood mercury levels. And they had a lower glucose levels, which was a surprise. And so anyway, you can run diet interventions and test the biomarkers, you know, so there's so much we could do. And we already published a paper in 2021 that gives you all the analytical methods for measuring mercury and lead children and with diet intervention guidelines. And that's in the World Journal of Epidemics. And so that's it for today.